screen.
Ok. So, começou aí, né, Márcio? Vou começar então, tá?
Sim.
Coloca também ele para compartilhar a tela, por favor.
Eu já coloquei.
Ah, tá bom, obrigada. Então, vamos lá. Let's introduce Domagoj Vucic. He's a staff scientist at the Department of Early Discovery Biochemistry at Genentech, a place that I miss a lot. His lab works in translating basic findings and signaling principles into novel therapeutics for the treatment of cancers and inflammatory diseases, especially by targeting replicase and IAP's ubiquitin ligases. One of his outstanding contributions is related to the development of IAP antagonists from molecular mechanism all the way through the clinical trials. More recently, his group and collaborators described the role of HIPK1 in tissue damage and in a number of inflammatory diseases, while at the same time showing that HIPK1 activity is not essential for tumor growth or metastasis. He's currently developing HIP1K1 inhibitors to treat these pathologies from preclinical models to future clinical trials. Domagoj is an international reference when it comes to development of therapeutic strategies based on cell death molecules. And because he's such a dear friend, he always makes time to contribute to our courses. Thank you very much again, Domagoj, and I hope that next time we meet in person here. Or there.
Definitely.
Yeah.
Thank you for the kind introduction. I would also like to give it in person, but this is our reality. So as you said, hopefully in the future we'll make it more enjoyable. So let's see.
Of course.
I'll bring up my slides. Let me know if you can see them.
Yes.
Yes.
Okay. Perfect. Okay, so I will start then. So thank you for inviting me. I'm happy to share our findings and perspectives on the cell death and targeting diseases that affect a lot of population in the world. Let's see, how do I advance? Okay, so it's been described many years ago now, 50, the terms of apoptosis and how cells shrink, chromatin compacts, DNA is cleaved. All that is really properly organized, and it's not a random process when cells are programmed to die and then they're gulped by neighboring macrophages. Mostly this apoptosis is relatively clean. There is limited inflammatory response. There is some, and it's really critical for life. You have during embryogenesis process of apoptosis, during the maturation of the immune system, regeneration, and so forth is really critical. However, on the other hand, deregulation of apoptosis and other forms of cell death can have pathological consequences, because if you have a little cell death reduced, you can go to cancer. And first part of my talk is going to be focused mostly on that. However, excessive cell death can cause degenerative diseases, inflammatory diseases, and that's going to be the second part of my presentation. Okay? In general, what it looks like when you would see that over time in microscope, for example, you see the cells nicely live in these layers, properly organized. But then one cell, such as this one, receives the signal to undergo cell death. Then all these molecular processes start, but then also external phenotypic processing starts. You see blubbing of the membrane, and then chromatin gets cleaved, and then these pieces are formed in the apoptotic bodies when you have pieces of the chromatin engulfed by the pieces of the membrane and other cellular content, and they are then suspended in the surrounding area, which would be really bad for the surrounding tissues because it can promote damage and inflammation. However, then we have specialized cells such as these macrophages, then engulf all these pieces and provide healthy and clean environment for the rest of the cells. Okay? And so molecularly, as you probably heard during this week, there are different ways to trigger apoptosis. So you have death receptors that can be triggered either by TNF, tumor necrosis factor, the best known, that assembles complex here, and then you have multiple adapters like a TRAD and FAD and TRAF2RIP1 that lead to caspase recruitment activation in a cytoplasm. Or you have death receptors such as trail receptors or fast receptors that really rapidly assemble this complex with FAD and caspase 8 and lead to activation of caspase 8. On the other hand, traditional way is through chemotherapeutics or radiation that's been done for last century in cancer patients, and that mostly leads to various activation of the pathways, but a lot of that focuses on mitochondria, and in mitochondria causes disruption of mitochondrial integrity, allows the cytochrome C to leak out of mitochondria in the process that's regulated by BCL2 family members, such as BCL2, BCLX that block that, and the others, as I mentioned soon, enhance this. And that cytochrome C, when it leaks out, binds to APAF1, and then that assembles a poplosome where caspase 9 is activated. And now we have active caspase 9, and both active caspase 8 and 9 then can converge on the activation of caspase 3 and 7, and then they cleave hundreds, thousands of cellular substrates and cause apoptotic cell death. And process that's so critical is regulated at many levels. For example, as I mentioned, BCL2 proteins regulate here mitochondrial pathway. IP proteins can block either TNF-mediated cell death, or XIP, X chromosome link inhibitory apoptosis, can directly inhibit caspases 3, 7, and 9. And then, of course, there is a amplification of that loop. So there is a second protein that leaks out mitochondria. It's called second mitochondrial activator of caspase, or SMAC, that then binds to IPs and prevents them from inhibiting these processes. And so first I'm going to focus on BCL2 because that's the best known and the oldest described factor. And it's really important in cancer. And when we look at the critical properties of cancer, in even this first review by Doug Hannah and Bob Weinberg describes several critical properties. And one of them is resisting cell death, because when tumor overgrows and proliferates, there is a lot of pressure to undergo cell death. However, the anti-apoptotic proteins that block that and that way allow tumor to progress and survive. So BCL2 is the first identified regulator apoptosis. That was many years ago, and it was identified because of this chromosomal translocation in follicular lymphomas. And then other, such as David Vo and others, described that this is really causing enhanced survival, not proliferation. Because before it was known that RAS, MYC, and others really work by enhancing proliferation. But the important part here is that you're enhancing survival of the cells, not just proliferation, which is a consequence of that. And we know that BCL2 is now frequently upregulated various mechanisms in hematological malignancies, in CLL and so forth, mostly because of the upregulation of its levels due to this translocation and enhanced expression. Okay. So BCL2 proteins comprise of two major families, pro-survival and pro-apoptotic. And pro-survival are BCL2, BCLX, A1, BCLW, MCL1. And proapoptotic can be either this BAX family, BAX, BAC, and BOC, or BH3 only. So all of these family members are defined by BH motif, BCL2 homology motif. And some of them have several, and some of them have only one, BHD3, which is critical for activation of cell death. So this apoptosis really requires BH3, and even just having BH3 only will allow these proteins to trigger cell death, okay? And so we have a lot of them, as you see here. And so why is that? Well, that's because different stimuli activate different members. So you can see for some of them, you can have a DNA damage or hypoxia for P53 activate some of them. Some of them get activated via various kinase inhibitors or chemotherapeutics. Some of them by cytokine deprivation, or some of them by metabolic changes in the cell or environment, or epigenetic changes, and some even by death receptors, so amplification loop. So that's why you have several members, because they make sure that every different tissue damaging signal is interpreted properly in a timely fashion, and then they then trigger activation by going to this BCL2 family anti-apoptotic proteins mostly, and block their activity. So you have transcription, sequestration, phosphorylation, proteolysis, all kinds of way of activating these proteins. Okay, as I said, they are not all the same, and some of them target just once, and NOXA just targets MCL1, and BAD only 3 of them, BIM and PUMA all of them. So it's really very dynamic, and you can see here that if you have these proteins, BMF, BAD, BIC, and so forth, you don't see much damage. You don't see much loss of cell viability, because they don't target all the proteins of the anti-apoptotics. However, BIM and PUMA target all of them. So you have them expressed, then you see massive loss of cell viability, because they can take out all anti-apoptotic BCL2 proteins at the same time. Okay, and structural studies were critical in defining these processes, because we know now, there is this BH3 motif goes into the groove of the anti-apoptotic BCL2 proteins, like just BCL-XL, and bind, and that's how it causes the whole activation of cell death. So schematically, if you think about it, you get a death signal, and then BH3 proteins will then block anti-apoptotic BCL2 proteins, which hold BAX and BAC in check. There is also direct activation. Some of them can directly activate, such as BIM, PUMA, and BIT, can directly activate BAC and BAX, but a lot of them act by inhibiting inhibitor in a way, okay? And so then what happens, you can imagine if you have this BH3 mimicking moiety, and you will block this BCL2 anti-apoptotics, then you can activate directly BAX and BAC. They will undergo conformational change, oligomerize, and now they can cause mitochondrial damage, caspase activation, and apoptosis. So this is really premise for developing drugs that can target this process and activate apoptosis in a BCL2 manner. Okay? And so the effort here was really to mimic the binding of these BH3 motifs, peptides, and how they bind to anti-apoptotic BCL2 proteins, right? And so there is this amphipathic helix, and there are several residues you see here indicated that contact this in this groove, in this shallow groove, and that they're critical for this activity and induction of cell death. So the effort was made, and initially by Abbott, and then later a Genentech joined, and we also with Rehive. So we had a trilateral collaboration between Abbott, Genentech, and Rehive to develop this BCL2 antagonist. And Abbott led it in a way because they had a more advanced chemistry and really developed several of these reagents. Okay, and first of them, the NATO clinic is ABT263 or Venetoclax. Venetoclax was mimicking BAD. Okay, and so it had high affinity for BCL2, BCLX, or BCLW, just like BAD. Okay, it was very efficient, low nanomolar or sub-nanomolar activity for all these proteins. Structurally made a lot of sense. As you can see here, this was not derived by classical screening, but by NMR labeled multiple pieces. So one small piece, one side, another small piece bound another side, and then they connected that. And that's how you got this drug, right? So single pieces would not sufficient, but when you combine it, then you have this nice drug that can then cause activation. And it's definitely defining Lipinski rules because it's a little bigger than traditional small molecules, but still very efficacious, okay? And so now this was taken to clinical trials, and we see that it was very effective, a lot of efficacy in BCL2 dependent malignancies, in, for example, CLL, where you express high level of BCL2 and BIM, it would disrupt this process and cause massive cell death. You can see loss of lymphocytes in the CLL patients, reduction in lymph node size, and all of these parameters really very good. In a lot of patients have very good responses, 90% of them in lymphocytosis, almost 40 in reduction of tumor volume. So it was very good for that matter. However, it was almost too good to be true because there are problems with this approach. And that is that David Huang at WeHi and colleagues discovered that there was a severe loss of platelets, okay? And the beginning was not clear why is that. But then through his studies, and you can follow with these papers, it was found that it's really BCL-XL is critical for survival of platelets, okay? And so when you hit with Venetoclax or with the ABT737, another compound, you rapidly see within an hour, you lose platelets, and in dose-dependent fashion in patients with Venetoclax, you lose these platelets. And that was definitely a dose-limited toxicity. Because it was efforts were made to maybe time it differently and to reduce the dose, but then you would lose the efficacy, right? And so definitely, and unfortunately, Venetoclax could not be continued in clinical trials, okay? And so now this is the problem. Venetoclax was targeting both BCL2 and BCL-XL. And then it was clear for a lot of these basic research events that is really BCL-XL is critical for this thrombocytopenia. But if you want to go to B-cell lymphomas, leukemias, lymphomas, you really need to target BCL2, and you want to avoid BCL-XL. And so then the hypothesis is if you have BCL2 selective inhibitor, you will maintain efficacy in heme malignancies, and you will not induce thrombocytopenia, and you will have better therapeutic window, and you allow safe dosing of cancer patients, okay? And so this was the effort that underwent with Abbott, later called AbbVie, and Genentech. And the key is that it was very difficult because they are similar. There are really just only 4 residues maybe that are different in this binding groove. And so for a lot of effort, few years and hundreds of compounds that were made, then we got from changing various moieties and parts of this molecule to find one that's going to have less BCL-XL affinity and still maintain BCL2 affinity, and with still have this binding conformation to bind to BCL2, but to lose thrombocytopenia. And that was resulted in a development of Venetoclax, which is a selective BCL2 inhibitor. So you went from this Venetoclax to Venetoclax. As you can see, you improved over 100-fold functional selectivity. So you see BCL2 versus BCL-XL, how you move the selectivity. But at the same time, you do not want to reduce efficacy in B-cell lymphomas. And so it's still active in B-cell dependent tumor cell lines, but ineffective in BCL-XL dependent tumor cell lines, okay? And so, and then lower affinity for MCL1 as well, okay? It was orally bioavailable, so it will be given as a pill, and all had very good properties. And so then the clinical trials were then continued with this compound, okay? And this, I'll just show you some of the preclinical data. Why was so good this compound? Because you have very good diagnostics, which is when you see BCL2 express high or moderate levels, it's good. but you have to have BIM at low level. If you have BIM at high level, then you already inactivate BCL2, you would not have much effect here, such as in Toledo. But in lines that you have low BIM and moderate to high BCL2, then you have nice activities. You can see here in green and is Rituxan, and in blue is Venetoclax. Very nice activity. And when you combine them, very good complete tumor inhibition in several of these models. So this is the major advantage here, that you had diagnostic tool, you can really screen patients, and by the profiling BCL2 levels, you can say which tumor is very likely to respond and which one is not, okay? Of course, now we are facing with a resistance, and some of them grow other factors, for example, MCL1, which sometimes gets overexpressed as a result of that, but at least we know initially which tumors to target. Importantly, as I said, this Venetoclax did not lose potency. It's actually twice better than Venetoclax in the CLL samples, but it's almost 100-fold weaker for platelet killing. So it really allows you to have this therapeutic window when you can still kill B-cell lymphomas, but you will not damage platelets, which was really critical, a safety feature to allow that to be approved. And then very nice effect in various tumor studies, CLL, you see lymphocyte count goes dramatically down, lymph node size down, bone marrow infiltrate, all that is reduced in vast majority of the patients. So very nice efficacy and very good safety profile that really allowed Venetoclax to be approved in a variety of these indications. And we are still doing more trials to advance more tumor types where Venetoclax can be approved. And so in 2016, FDA granted approval for Venetoclax, and that's really the first drug, the first treatment that's based on cell death and apoptosis that's approved for the cancer treatment. Okay? And later that year, we had approval in Canada and then in Europe, all over the world, and actually got the reward as the best new drug of the year for that time when it was approved. And as I said, there are multiple trials going on to advance it in various different types of tumor. Okay? So I can say that when you look at this, the schematic and many aspects, and there are efforts, I can tell you that thousands of biopharmaceutical companies that are targeting cancer, various aspects. At least here, we have shown that it's possible, because Venetoclax is a selective, potent, orally bioavailable small molecule with all these features that I described. And it really serves as a great proof of concept that you can really target cancer by going after cell death and inducing cell death in this compartment where there is insufficient apoptosis. Okay. So this was a first effort, which makes sense because that's one of the oncogenes, BCL2, that was among the first ones to describe, and there is massive effort. There is still effort now focused more on MCL1, because, for example, for the hematological malignancy, BCL2 is critical, but in the solid tumors, not so much. And now there are several companies that are developing MCL2 inhibitors, either to inhibit it or degrade it, via PROTACs and other efforts, how to eliminate MCL1, and that way really expand the number of tumors, especially solid tumors, that can be targeted by this induction of apoptosis principle. Okay. So the other efforts that we're going over are pro-apoptotic death receptor agonist or SMAC mimetics or IP antagonist. I will, for sake of time, I cannot cover all of them, so I will not talk about death receptor agonist, but I will talk about IAPs. Okay, and so those are the other class of anti-apoptotic proteins that block apoptosis induced by death receptors and chemotherapeutics, expressed in many cancer cell types. There are 8 human members. They were initially identified in baculoviruses. Those are double-stranded DNA viruses that affect caterpillars, like a cabbage looper or butterflies. They're important for agricultural industry and forestry. Okay, And then later, through genomic similarity, they were found in various screens, they were found in all animal phyla. As I said, some of them are expressed in cancers such as melanoma, IIP, MLIP, that's really selectively just the melanomas and not the other tissues and tumors, as opposed to benign nevus. And functionally, there are a couple of domains that are important. This is this BIR, baculovirus IIP repeat. This is how they inhibit caspases. This is how they bind to IP antagonist and a lot of other protein-protein interactions. On the other hand, we have ring at the C terminus, and that gives them ubiquitin E3 ligase activity. And I will go over that in subsequent slides. So now we are going downstream. So we started here at mitochondria and BCL2, okay? And so now we see what happens after BCL2 is overwhelmed and you have the signal proceed. And then in this case, you have IIPs such as XIP holding in check caspase 9, caspase 3, and 7, okay? However, what happens? You have this ionizing radiation, chemotherapeutics, and then overwhelms BCL2, and then SMAC or Diablo leaks out of mitochondria. And what happens now? It binds IIPs, it takes it away from caspases, and now caspases are weaponized, they are armed, they get processed, assembled in lethal machines, and now they can cause apoptosis by cleaving all the substrates, okay? So you really taken away inhibitor and armed the caspases, and now they cleave. And so now the idea is to mimic this, to make small molecule mimic this SMAC mimetic and to get this IP antagonist to antagonize IP proteins and activate caspases, okay? So as I said, this is mostly the case for XIP, where holds in check caspase 9 and SMAC comes, you see with a similar residue, and then activates caspases by taking away IIPs. And again, this is very well evolutionarily preserved because we have the same thing in flies, in fruit flies, Drosophila melanogaster. You have here Grim Reaper, sickle, these proteins that were found there for genetic screens have the very similar regions. And it's really just this 4 amino acids, AVPI, that gets into this binding groove and allows activation of caspases. And now we have also structure of that. So we see it's a very nice pocket, and it's kind of very nice for the drug development to mimic binding of this peptide into the groove on the protein surface. Okay. So what was starting point was really the peptide, AVPI. And again, Genentech and many other companies, we did a lot of screening. Millions of compounds were screened in these libraries, and guess what? None of them were better than what nature gave you. Nature gave you this AVPI with this, low micromolar affinity, and it was still better than anything that came out of screen. So what we did, we basically did structure-based evolution of peptidomimetics. So we changed everything around this AVPI peptide. So eliminate peptidic for nature, make better solubility, permeability, stability, all that. And so eventually you end up with a small molecule that has improved binding affinities, but also improve all these other properties, pharmacological properties that maybe don't sound very sexy, but are critical for having a reagent that can be used in vivo and later in patients, okay? And so we tested that in many models. I'll show you some of the data. For example, these are the tumor models. One is the subcutaneous lung cancer A549 model. You let cancer cells, xenograft model, right? Inject in the flank of the mouse. When it gets around 200 square cubic millimeters, then you start dosing either with a vehicle or with your compound. Here is one of our clinical candidates, GDC152 and 20 mg per kg. As you can see here, vehicle allows massive growth. Within 2 weeks, it goes to the 1,000. However, if we dose with our compound, very nice inhibition in this case. Similarly, we did that with a orthotopic model. So orthotopic means you go with a lung cancer cell line and you inject into the lung, into the same tissue where it should belong, epithelium of the lung. And so you start now, again, let it develop for some time. Okay, so you see here in micro CT imaging, this is the pretreatment image in both tumors, very comparable size. However, you see after 2 weeks with vehicle large tumor size, and here dramatically reduced tumor size when you have IP antagonist treated animals. So this kind of data allow us to go to clinical trials, and there are actually several compounds that went into that process from different companies. Some were oral, some were IV, were monovalent, where you just have AVPI. Those are monovalent, but if you combine two of them with a linker, then we call them bivalent. They're too big to be orally available, and that's why they have to be IV, dosed IV. Not as practical, but much more potent, okay? And interestingly, what we observed then, that some of these compounds, especially bivalent, cause massive degradation, a very rapid degradation of CIP1, cellular inhibitor of apoptosis. It's just within minutes. Monovalent also, but less fast. Bivalent very quickly causes that. So I'll just tell you how we figure out what's going on here, because we didn't understand. With BCL2 antagonist, you bind to BCL2, nothing happens to BCL2. And here you degrade within minutes the target. So why is that? Okay. And this is not just in cells. We see that in vivo. We see now that tumor xenografts treated with the IP antagonists, also we see CRP1 degradation. And actually now all the companies that do clinical trials, they monitor CRP1 degradation, and that's the pharmacodynamic marker, PD marker. So you can say, okay, I do not know if I'm going to have efficacy or not, but if I see that CRP1 is degraded in PBMCs from a patient, at least I know that this patient has received efficacious dose to degrade CRP1. And that way we know that, okay, if we don't see efficacy in tumor inhibition, it's not because we didn't have enough drug on board. No, it's because protein maybe didn't matter for this tumor or wasn't dosed in proper way or something like that. Okay? So this brings me to ubiquination. As I told you that IPs are ring domain E3 ligase. So ubiquination is next to phosphorylation, one of those the most common and key modification. So that entails multiple steps. So you have activation with the E1 ubiquitin activating enzyme, then you have conjugation where ubiquitin moiety is conjugated, and then you have ligation. And this is where E3 play a role. E3 ligases select the substrate, and then E2s transfer ubiquitin to the substrate. And you have various ubiquitin moieties. You can have monoubiquination, and that's usually just in DNA damage, but often you have polyubiquination, and you have different conformation of different ubiquitin chains. So for example, if you have it K48, then you go to proteasome and you degrade the protein. However, if you have linear or 63 or sometimes 11, then you have complex assemblies such as NF-kappa-B, and then you get signaling activation or response to DNA damage and so forth. Okay, and so in this case, we know that CIP1 plays a role here and can ubiquitin multiple substrates such as RIP1, NIC, and TRAF2. And for some of them, like NIC, it's really degradative, causes proteasomal degradation and causes non-canonical NF-kappa-B activation. However, in the case of RIP1 and TRAF2 or itself, it participates in canonical NF-kappa activation and signaling complex assembly. Okay. But what is going on when IP antagonist hits the compound, the CIP1? So we realized after years of effort that there is a complex transformation that BR3, this is where IP antagonists bind, is actually in contact with the ring domain and keeps it in check. So the ring cannot be active when BR sits there, okay? And then what happens is that it's in closer conformation, and the IP antagonist comes and opens it up. And then the ring can then dimerize and be active, and then it can proteasomally degrade substrates. And we can see here that if you have CIP1 wild type, see, it's fine here. You add protease inhibitor, you can stabilize further. But if you add IP antagonist, you degrade it. However, if you eliminate this contact and open it up from the get-go, then protein is always unstable, even without IP antagonist already degrades. So, And that is really critical for the not just tumor, but for the activation of many processes. So just to summarize this part so far. So you have TNF binding to TNFR1, you recruit CIP1. And then if you throw IP antagonist, this is what happens. Then ubiquination occurs and CIP1 auto-ubiquinates itself, okay? As a result of that, you're gonna have proteasomal degradation, but you're gonna have also activation of non-canonical NF-κB signaling. And as a result of this activation of non-canonical NF-κB signaling, you have production of various cytokines and chemokines. Among them is TNF. And TNF can now come whereas in pyrocrine or autocrine fashion, bind to receptor and trigger signaling. However, there is no inhibition anymore, and then you're going to lead to apoptosis and cell death because their inhibitors are gone, they're degraded. Okay, and so that's really premise how TNF can cause cell death. Only when you eliminate IAPs or other NF-κB regulators, then TNF can stimulate apoptotic cell death. Otherwise, it's really just proliferative stimulus. Okay, so, and this is just to show you, this is not just one example. There are many proteins. For example, BCL2, remember MCL1 is often degraded. P53 is degraded. There are many ways and aspects and places where ubiquination plays a role in regulation of apoptosis, because when you regulate the levels, then you can really say if protein is available for in protein-protein interaction or not, and where protein is at the right place at the right time, all can be regulated by ubiquination. However, how it regulates non-canonical signaling. So here is the deal. If the CIP1 binds TRAF2 and TRAF3, and they are in complex with NIC, okay? And when NIC is NF-κB inducing kinase, and it's continuously degraded. So it's ubiquitinated by K48 chains and is degraded, and you don't have activation of non-canonical signal, okay? However, what happens when you get IP antagonist? IP antagonist comes here, binds to CIP1, and now CIP1 is gone. And now NIC gets upregulated, and now you can get processing of P100, first IKK alpha phosphorylation, IKK alpha then phosphorylates P100, which undergoes partial proteasomal processing to P52, and with rel-B goes to the cytoplasm, and you have inflammatory cytokines. And similar principles are used by many TNF family members, TWEAK, CD40, CD30, LIGHT, and so forth. As you can see here, all of them involve, just with IP antagonist, degradation of CIP1. So when you have CIP1, NIC is not present there. You see, there is no NIC. However, if you degrade CIP1, then NIC can show up, and then you have processing of P100 and so forth. And so why is that important? Because I'm telling you all this because actually we tried IP antagonists in several different tumors and different trials. There was very little efficacy, and we could never find out why some tumors respond and some don't, because there is, all of them have CIP1, all of them degraded, but it's not clear why. And so we believe that actually activation of non-canonical signaling could be the key here, because if you provide this inflammatory environment, then you can combine that with checkpoint inhibitors, such as PD-L1 antibody. And this was shown in this nice paper from a few years ago, that IP antagonists such as LCL161 by itself can promote survival in these mice that are carrying tumors. However, and in this case, PD-1 antibody provides very little benefit. But if you combine it, very significant benefit in reduction in tumor growth and very significant benefit in survival. So that means when you induce degradation of CIP1 and to release NIC, activate non-canonical NF-κB, then you produce this immune activation, and then you can have anti-tumor phagocytic activity and nice combination with checkpoint inhibitors such as PD-L1. And there are actually combination trials like this ongoing, and I think they have much better chance of showing some effect in tumor trials, okay? The other application of IP antagonist that was not apparent right away is in the viral infection. and this is with the HIV latency. Because HIV, although now we have very good efficacious treatments for HIV, with the cocktails, protease inhibitors, and so forth, actually people can live for many, many years with HIV, but they never get rid of it because HIV goes into latency and is dormant and they live with it. The idea here is that if you activate non-canonical NF-κB by IP antagonist, you will break the latency, okay? So IP antagonists can disrupt, activate non-canonical signaling, and that will break the latency and promote active viral replication. And now you hit it with a cocktail of drugs, and then you can completely eliminate it. So that will be, you reverse the viral latency, and then you hit it with a cocktail, and then that way, hopefully, you can for good eliminate viral infection and free people from HIV for good. So we are engaged, we have collaborators with this effort, and hopefully we'll show some promising results in years to come. Okay. So this kind of concludes this part of my presentation, where I talk about targeted therapies, describe how beach mimetics really made it into clinic, and we have now drugs, and we're still working on SMAC mimetics, IP antagonists, to see if, but not by themselves, but in combination with other checkpoint inhibitors, or maybe in some viral infections, and we can find application for it. Okay. So I can pause here for a minute, and if there are any burning questions, I'll be happy to address them.
All right, Domagoj. Students, anyone with questions for Domagoj now? Lucas.
Hello, everyone. Are you listening to me?
Yes.
So I got a little curious when you talked about Venetoclax and the treatment for CLL, because recently I'm reading about it and I saw that a few patients are suffering from resistance to this drug. So I would really like to know if there are some data trying to use those EPA antagonists on CLL, if you know about it.
To use which IP antagonist you mean, or?
Yes.
Yeah, yes.
So I'm not aware of those studies. There are no clinical trials for sure, but I don't know if there are preclinical trials on that. I think the major effort there is really to see what are the resistance factors. And I think one of the major resistance factors is MCL1, okay? MCL1 gets upregulated in this situation, and now there are efforts to target MCL1, as I said, to inhibit it or to degrade it, and that way to eliminate. There were some preclinical trials, studies, experiments in mice where they combined SMAC mimetics, IP antagonist, and Venetoclax. And in some cases, there is some effect. In some cases, there is no effect. It's not clear what's driving resistance and if IPs are really the major resistance factors in that case. So I'm not sure how that will proceed, and I'm not sure. I know that there are no clinical trials, but I think resistance there will be really, for Venetoclax, resistance will be more focused on other BCL2 family members.
Yes, I got it. Thank you. Actually, what I really read about it is that the resistance was because mutation that the BCL protein suffered and couldn't attach to the drug anymore.
Oh, there you go. You see, so that's the thing, that the tumors, you know, people do these experiments in labs, and you have tumor cell line, and you put a drug, and you keep it, and only 1%, 5% will survive. After a month, you get a population that really outgrows, and it has all kinds of mutations. And this is not just a cell line. You can go to patients, and you see really, after a month of treatment, patients also develop, just like in cells, they develop mutations in key proteins, key genes, and that leads to resistance and tumor progression. So luckily now, with advancement of sequencing, it can be done much faster. So you can, when people don't respond, they undergo exome sequencing, and you can see what is going on there, why at least those critical genes can be analyzed to see if different approach should be applied.
Domagoj, can I just jump in quickly? Normally, as far as I understand, these target therapies with the BH3 mimetics is given like constantly. Is there any work or trial as to kind of like pulse and chase and like combine it with something else, and then, you know, just to avoid this sort of a pressure?
Yes. Absolutely, absolutely. All that is done, and you know, they call it drug holidays. So yes, patients would go on a treatment for a couple of weeks, then 1 week off, then put them back and try combinations. All that is done, and I mean, that will be totally new, different presentation just to see clinically how you do that, because there are a lot of things done there, combined with sequencing and so forth. Yes, absolutely right, absolutely.
Somebody else with
Gustavo, I think. Gustavo.
Oh, there is a cat that has a question.
This is Karina's cat. You know, I want to make a cat for dinner.
Okay. There'll be very little meat, but okay.
You know that, Domagoj, just for the sake of the break, you know that if you go to watch football games, you will see a lot of these charts with beer and some sticks, you know, with meat. So these are all cats.
Wow. This must be only Rio de Janeiro fans.
No, I think it's everywhere. You buy it thinking that is beef, you know, is like cattle, but it's
or rabbit, yeah, but it's not.
I think there's another question.
Okay. Gustavo Martins.
Hello.
Go ahead.
Can you guys hear me?
Yes, I can hear you now.
Yes.
I'm sorry, I won't be able to turn on my camera because I actually don't know why. It's kind of malfunctioning somehow, but thank you, Professor, for the lecture. I believe we are going to study a little bit more in the context of autoimmune diseases or degenerative diseases that you said. Mm-hmm. And I during all your presentation, it kept me thinking, and I didn't know if I should ask it now or at the end of everything. But here in Brazil, I believe that we have a struggle to research good science, and we are always trying to improve our science. But one thing that we still lack is the participation in the pharmacological industry and the ability to develop new drugs, especially here in Brazil, in the context of Brazil. Attending to your lecture now, it kept me thinking what is the cost of all this new drugs development in terms of time, in terms of money? It is something in other world. It is something much different from our reality. How is it?
Well, it is expensive. I can tell you ballpark, approximate price, what it costs in US to develop drug. It takes usually 10 to 12 years, and it would cost you a few $100 million. Preclinical trials, preclinical studies would cost $10 to $20 million. Phase 1 would be also 10 to 20 million. Phase 2 would be 30 to 70 million. And then phase 3 can be 100 to 200 million dollars. You know, in phase 1, you're treating maybe 30 healthy volunteers. In phase 2, you go up to 50 to 100 patients. And then in phase 3, we're more expensive. Then it's going to be completely randomized. Not even doctors should know, or patients should know who gets placebo, who gets the right drug. Multiple centers, multiple monitoring, takes several years, and that will lead to ultimate approval, and that really can cost $100 to $200 million, maybe more, right? And so it is extremely expensive, labor intensive, and long-term. And I can tell you that the chances of developing drug are very low. If I tell you that one compound, every compound that even goes into development, when I say development, that means it goes from basic research into the phase where you're going to see toxicology studies in rodents and in monkeys. Okay, that kind of drug will then, if it passes, go to clinical trials. It has 5% chance of success, 5% only. Okay, so 1 out of 20 will succeed, and they will fall off. And earlier they fall, it's better because you save money. The worst thing is when you have failure in phase 3. That's what you definitely want to avoid. So earlier you can eliminate, that's much better. And most elimination is because of the safety, because you can always find something to bind. You can find efficacy in your preclinical models, but to know who you're going to treat, so patient selection, diagnostics, and to be safe, those are 2 key factors that are most difficult and take long time and money to develop. And so that's why in the countries that are not rich, in countries where you lack a free flow money, it's very difficult. And I can tell you that in US, I think because of the environment of venture capitalists and a lot of the recent internet money, the funding is not so difficult. I'm serious, it's not so difficult, because and its understanding is that most of the things will fail, but the one that makes it has to cover for the cost of many others. So that kind of accounts also for the price, because you cannot, from one drug should not just recover price of its development, it has to recover price of 10 others that failed along the way. Otherwise you won't have drug. The hope is for the, and I'm talking here, I just want to specify, I'm not talking as my company representative, I'm talking as a person, a personal, my individual perspective. And that makes it really difficult for many countries in the world to develop drugs, and many countries in the world to have access, rapid access to the drugs that were just approved and got on the market. So you have to wait, I don't know, 15 years or more for the patent to fall off, and then you can see, okay, now we can use it and manufacture it cheaper and so forth. There's also a lot of drug repurposing. So there are drugs that were tested in one kind of disease, but actually can use in many others. And so there are many efforts there. And I think with time, the price will go down, and with the sequencing of all these people genomes and the tumors and inflammatory diseases we're going towards more personalized healthcare, just like with BCL2. And now in the second part, I'll show you also example how we are trying to do that in inflammatory diseases. So I don't know, it's a long answer. I don't know if I answered your question or not.
Yeah, you answered it perfectly, and I am also astonished by in terms of time, how long it takes. And you said it takes maybe decades, 15 years, and
yes,
it's actually really interesting.
Yeah, you just think about it. Even phase 2, you want to treat 100 patients, you have to go to 5 to 7 hospitals, you have to enroll patients. That's going to take 2 or 3 years in a very good situation. With pandemic, everything kind of froze down, but that's going to take just 2 or 3 years. And phase 3 is going to take 4 or 5 years to get all this randomization and properly evaluation and everything. So it is a long-term process.
Can I just
add?
How
many? Excuse me, Gu. I missed. How many patients normally you have in the phase 3? Millions.
Well, it depends. It depends from the trial to trial. For example, if you want to go to some cancer that's not very common, okay, then you can maybe go with 100, 200 patients and have it approval. But if you want to say, oh, I want something for cholesterol reduction, okay, or something that's going to help with a heart attack, then you're talking about thousands and thousands of patients, okay, because you have to have statistical value and improvement over standard of care. So it really depends from disease to disease and from reagent to reagent.
Okay, thank you.
I just want to add a couple of things. One related also to our reality here. The problem in Brazil is not only about the money, but it's about all the laws that make it difficult for a company to patent. Yeah. So we have so there is no incentive for, you know, somebody to invest money, such, you know, amount of money, if then the patent may be broken in Brazil. So and this is a problem that I think we should think about it, you know, very careful. Because
no, it's complicated. I think in India, similarly, also I think is that patents don't matter much, or you know, China government controls all of that. So you know, it's very different in different parts of the world. I'm familiar with the Western Europe and US, Canada. Those are the markets, and that's when we think about the profit, we think about those markets. Everything else is kind of a little extra. If it happens, happens. But these are the main markets that you can enforce patent protection, enforce you know, infringement rights and all that.
And in Brazil, the trouble with bureaucracy and companies, it's not only related to pharmaceutical medical companies, is everything. So I just want to you know, say that because we've been suffering from criticisms during the pandemic related to the amount of money of these pharmaceutical companies, all this shit that happens in politics are being brought to the medical scenario, okay? Yeah. And I think we scientists should keep a very rational perspective. Mm-hmm. So at one point, we need to understand, as Domagoj said, that, you know, there was a huge amount of investment. So they need to make money, the companies, otherwise there will be no more investment, and then there will be no more drugs to attend the population. So it's just a bit ugly and small, to my mind, to, you know, put this on the agenda. No. And the second thing that also related to that is, once, as Domagoj said, there's a lot of things that can be repurposed, the drugs. And some is not really the drug, but the platform, as we see in vaccines. So the vaccines, some of the vaccines now takes not that much longer, because it already took 20 years to establish a vaccine platform that can be just tweaked a little bit. So these are things that you guys, students in health science, should keep in mind to, you know, fight in your WhatsApp groups again, all these bullshits that invade our, you know, or disinform us, you know. So I think Gustavo's question is very, very important.
Yeah, and I think it's often, you know, difficult to get this information, and because it has to come from kind of independent body. But I'm talking from my experience being over 20 years in this industry and see how it goes.
Excellent. Thank you,
Yes.
Domagoj.
All right. So
Thank
should I continue to the last part?
Yes, you
Yep.
should.
Okay. Well, so now we're going to go a little bit into inflammation. Not a little bit, a lot. So what happens when you get infection or tissue injury? You get this low-grade inflammatory response, and often you have pathogen clearance. We have all these inflammasomes. We have all these tissue repair mechanisms, and you go to normal tissue homeostasis. However, if you have persistent stimuli, over time you can get to tissue damage and release of DAMPs and cytokines. DAMPs are danger-associated molecular patterns, and that will cause amplification in inflammatory response, recruit macrophages, lymphocytes, and so forth, right? And so this is a process that's active in many inflammatory diseases, such as inflammatory bowel diseases, arthritis, psoriasis, and so forth. And we believe there is need for therapeutic intervention in these cases. And you can tackle it from many perspectives. That what I'm talking about is cell death based and tissue damage based approach. Okay. And again, we're going to go back to cell death. And I described so far apoptosis, how we have this cleaner form of cell death, core components defined. We have drugs like Venetoclax here, but there is another form of cell death called necrosis, and that it was believed to be default pathway. But now we know there are various forms there. Physical, ischemic, or inflammatory. And inflammatory can be either pyroptosis, which means caspases 1 and 11, or necroptosis, made by kinases RIP1 and RIP3, mostly when caspases are inhibited or not activated. And I'll talk mostly about necroptosis and interaction with apoptosis in context of inflammatory diseases. Okay. So first, just briefly to give you perspective from how we studied this. So in this case, again, TNF gets activated by TNFR1 binds TNF, and then you have this complex CIP1 ubiquinase RIP1, and you get NF-κB mechanism activation. And protein like NEMO binds here, participates in this. On the other hand, deubiquinase like A20 blocks that process and prevents this. However, over time, especially when NF-κB is not fully activated, RIP1 moves into cytoplasm, and then with fat can lead to caspase binding activation, and then you get apoptosis, as we talked about. However, if caspase 8 is not fully activated or is inhibited, then RIP1 can auto-activate, goes to active conformation, autophosphorylate, then binds to RIP3. RIP3 also autophosphorylates, and now RIP3 can bind and phosphorylate pseudokinase MLKL. Pseudokinase means that it has domain that looks like kinase, but it's not active kinase, doesn't have activity. And that leads to necroptosis in a process that's not just defined by TNF, but tissue damage or toll-like receptors can also activate that. We see that in context of kidney injury, stroke, heart attack, also activate this process and lead to apoptosis, necroptosis, eventually causing membrane permeabilization, release of DAMPs such as IL-1 alpha, HMGB1, and so forth. And this is an active process, not just linear going one way, because there is a lot of crosstalk between various forms of cell death, and even with pyroptosis. But I'm going to focus just on apoptosis and necroptosis for the remaining of this presentation. Okay. And so first, we said, let's block necroptosis. Okay. So this is bad. It's causes tissue damage. And so RIP3 is really kinase that's active here. So this would be the best target, right? Well, guess what? It's not. And that is because it's toxic. And it was a very surprising find that Kim Newton and Vishwa Dixit made several years ago. So they made a kinase dead version of RIP3 because RIP3 knockouts are alive. But RIP3 kinase dead is not, because all of a sudden it switches. Instead of necroptosis, it binds RIP1, and that allows activation and binding caspase 8 and causes apoptosis. Okay? And actually, GSK, our competitors, made kinase inhibitors of RIP3 and also show toxicity. And so it was shown genetically in embryos, in adults, by chemicals in all different ways. There is activation of apoptosis and toxicities that prevents RIP3 from being a suitable target. However, RIP1 is not. And this is a great story and paradox of this field, that if you mutate RIP1 kinase activity, then you will block necroptosis. As you can see here, you block necroptosis cell death, but the animals are fine. So if you have RIP1 knockouts, they are dead, they are perinatal lethal. But RIP3 knockouts are viable. Opposite, if you just mutate one amino acid, it's just one amino acid. Now RIP3 kinase dead knockins are dead, but RIP1 kinase dead knockins are viable and completely fine. Okay, and so this was surprising, unexpected, but just tells you how sometimes you need to make these genetic models to verify, is this target good? And just knocking it out is not good anymore. You really have to eliminate critical functionality, and then you can verify if it's going to be good as a target or not. Okay. We further verified that in TNF-driven sepsis model, hypothermia. So you put large amount of TNF, then animals die. And if you have RIP1 kinase dead activation, you see they all survive. And you have RIP3 knockouts, they all survive. However, if you have RIP3 kinase dead, they'll be dead from the get-go. So this is the models we use: RIP1 kinase dead or RIP3 knockouts to verify various aspects of this signal. Okay. And then we also developed RIP1 inhibitors. So this is one of our critical tool compounds called GNE684 that has nice potency in human and mouse. RIP1 biochemical and cellular. It has very good pharmacological properties. This structure binds nicely here. Very selective, doesn't touch any other kinase. And we use this modified Sears model where we combine TNF and ZVAT. So instead of waiting for 12 hours, we just need 4 hours. In 4 hours, all animals die. And then you see compound here in dose-dependent fashion nicely inhibits this hypothermia and allows survival of the animals. Okay. So importantly, as I said, we did all these studies with RIP1 kinase dead mice to show they have no problem. So you see TNF activation of NF-kappa B, JNK, ERK, no problem in the wild types or RIP1 kinase dead mice. We look at the lymphocyte subsets, no difference. They're completely okay in wild type or the kinase dead mice. And also, if you look at the compound, unlike IP antagonist, no effect on RIP1 levels. Even if we go to 200 micromolar, no effect, and no effect on TNF-mediated activation of A20 or CIP2, which are early NF-kappa responsive genes. So it just tells you it's safe to target RIP1. There is no immune problems. There is no proliferative issues, none of them. Okay? However, still, where to go with RIP1? Over last decade, there are hundreds of papers, maybe more, that show implication of and involvement of RIP1 in tissue pathology in various diseases, acute diseases, kidney injury, heart attack, stroke, in cancers, in male reproduction system, in viral infection, in many chronic diseases, IBD, psoriasis, arthritis, pancreatitis, you name it, in many neurodegenerative diseases, ALS, Alzheimer's, MS, stroke. And so we really wanted to investigate using genetic models and tool compounds in the animal disease models, where does the RIP1 really matter and its kinase activity, and where we can safely and efficaciously inhibit it. Okay? So, for example, there was a Cell paper showing that RIP1 plays a role, or RIP3, in pancreatitis, and there was a fact of RIP3 knockout in pancreatitis model. However, we could never reproduce that. We see no difference between wild types or RIP3 knockout mice or wild types or RIP1 kinase dead mice in the serum amylase in this kind of serolin-induced model of pancreatitis. So we discounted that. Then there was a lot of papers about talking about the real role of RIP1 in tumors, that RIP1 inhibition reprograms macrophages, and that can be combined to cause this immune environment and combined with PD-1 antibody and so forth. Also that there is effect on metastasis. And we were like, wow, is this really true? We really need to investigate this. So we took the same models that were used in this Nature and Cancer Cell paper, okay, and tried to reproduce that. By the way, in that time, GSK, our competitors, tried to start the clinical trials in tumors with RIP1 inhibitor. So we were thinking, well, you know, as I showed you in the apoptosis pathway, it was a lot of things were tried. Death receptor, then and p53 targeting, BCL2 antagonist, IP antagonist, and we know now there are a couple of clinical compounds and there is approved drug. But what about RIP1 and RIP3 as a targets in necroptosis and tumors? Is it possible? Well, we went to the as endogenous, as physiologically relevant model as possible, which is this genetically engineered mouse models of pancreatic cancer, which are either driven by KRAS, P16 mutation, or KRAS, P16, P19, and P53 mutation, okay? So these mice spontaneously develop tumors after several months of age. And then when they were developed tumors, then we start treatment with compounds. As you can see here, there is no effect on survival. There is no effect on tumor growth. And we did that for a couple of models and couple of compounds and never observed any effect in this case, okay? Even when we crossed RIP1 kinase dead mice to this genetic models, again, there was no effect whatsoever, okay? The second idea was that there is this macrophage reprogramming that RIP1 inhibition by itself can cause phospho-STAT1, okay? And change the expression of different genes in macrophages. So we have looked at that many times. There was no effect. Kim Newton looked at that in kinase dead mice, no effect whatsoever. And then we also see that our Genentech compound or GSK, we made GSK compound. You can see here, very similar potency in mouse cells. However, there is no effect of STAT1. Only when you throw interferon beta, then you get phospho-STAT1 as you should, but nothing by RIP1 inhibitors. And so we, published our result, and soon around that time, RIP1 actually inhibitor from GSK was pulled out. They never actually treated patients. They submitted application to do it, but at the end they didn't treat anyone, which is good for patients because this is definitely very wrong approach. Okay? So over time, we really, verified that RIP1 matters in kidney injury and in heart attack, but not in psoriasis, not in pancreatitis. It also does not matter in cancer, but it matters in forms of colitis and Crohn's disease. Okay. Also, I don't have time to show you, but we show that there is no role of RIP1 in vaccinia or EBV, viral clearance, or in male reproductive testicular degeneration. That's totally invented story, right? However, there are other questions here regarding neurodegeneration. So we want to explore that as well, okay? So the ALS, Lou Gehrig disease, affects a lot of people, and the golden standard model there is SOD1 transgenics. So these are the mutation that happens in few patients, maybe 3 to 5% of the human patients have mutation in SOD1. And so now the model is that SOD1 transgenic that mimics this mutation in patient can be used because it has some features of ALS. So we cross that to wild type mice or to RIP1 kinase dead. As you can see here, no difference in all these different aspects of axonal pathology, average lumen size, different parameters measured. There is no difference with wild type cross or RIP1 kinase dead. Similarly, inflammation in spinal cord of these animals by IBA1 staining, no difference in wild type or RIP1 kinase dead. So we published that, and we are not pursuing that disease. However, to do more study in neurospace, we made additional model, which is rats. So we made the rats that are RIP1 kinase dead knock-ins, okay? Again, just one amino acid substitution. So you can see here, again, lymphocyte subsets, okay, these rats live just like wild types. There's no difference, the same lifespan. They activate NF-kappa B and have RIP1 levels the same level. However, they are resistant to necrototic stimuli. Again, here, we treat them with TNF or TNF and ZVAD, and wild types die and lose temperature rapidly. However, if they're RIP1 kinase dead, there is no temperature loss and they all survive. Okay, so that way we verify that we have a model that we can use for neurological diseases. Furthermore, to do the stroke study, we first look at the brains of these mice. So we did micro CT scanning of brains of the wild type rats or RIP1 kinase dead rats. And there are no differences. When you look at this cerebrovascular anatomy, there is no differences. And so with that idea, then we go to the occlusion model, major artery occlusion model to get stroke in the animals. You block this for 45 minutes and you release it, and you later look at the various aspects. Critical one by CT is the lesion volume. As you can see here in the wild types, you have this lesion in red. However, in RIP1 kinase dead, much less, much reduced lesion volume. So that means they're greatly protected in this case. We also looked at the myelin stain. So you can see the animals that underwent stroke, wild types, a lot of loss of myelin. However, if the RIP1 kinase dead, that's almost all preserved. And this is quantification here. So it just gives you example how we investigated these diseases. As I said, describe which ones we believe are not similar. In ALS, we don't think, at least based on this limited data, is the right one. However, stroke, we think, is really one that is affected by RIP1 because, when you have ischemia of refusion injury, that's the best indication for necroptosis. And I think that's the place where necroptosis really plays a role, is in ischemia of refusion injury, either in kidney, heart, or in brain. Okay. So having investigated those, we actually now, I want to focus more on chronic indications. Okay, not psoriasis, but other skin inflammatory diseases, rheumatoid arthritis, and IBD forms. Okay, and in this case, the genetic models that we use are the ones that we see mutations in patients, but we have also mutations in mice. So, for example, NEMO, we have mutations in various aspects of Lubach, HOIP, HOIL, Sharpin, and so forth. And we know when you eliminate those or mutate those factors, then you activate RIP1 and you have RIP3 dependent necroptosis or caspase A dependent apoptosis. Okay. So first I'm going to show you rheumatoid arthritis model. We verify activation of the pathway in human tissue. So this is human tissues control or patients with rheumatoid arthritis. And then we use RIP1 S166 phosphoantibody. As you can see, very nice activation in rheumatoid arthritis of phosphoryp1 signal. And now, if we do this model of anti-collagen antibody-induced arthritis, you see here that RIP1 kinase dead animals have 50% reduction in the clinical arthritis score. It's not 100%, but it's 50% reduction. And then we repeated that with other compound or with TNF blocking reagent. As you can see, remarkably, compound has the same effect as a RIP1 kinase dead mice, right? 50% reduction here, 50% reduction here. And TNFR2 also blocks the same way. And when we combine them, there is no added benefit. So it just tells you in this model, whatever TNF blockade does, RIP1 inhibition does the same thing. And combining them is not going to give you any extra benefit. Okay? The other model we explore is the Sharpin mutation. It's called Cpdm mice. Okay, as you can see in psoriasis, very little phosphoryp1, but there are other, like the diseases where it's more cell death, that we have more phosphoryp1. And so we took Sharpin mice. So these mice develop spontaneous skin inflammation after 5 to 6 weeks of age. And so we waited for that long, and then we treated them. This was a pilot experiment, and we treated them only for 4 days with a compound. As you can see here, after just 4 days, you see still remarkable reduction in their inflammation in skin, in dorsal skin and ventral cervical skin. You can see here, this is the guy that got compound. This is the mouse that got just vehicle. So definitely much more inflammation in this case. So we said, okay, well, if we did just 4 days, let's try 4 weeks. So we did now with 4 weeks, and we see now very complete protection from this skin inflammation this is histology. This is with a compound looks just like a wild type. They didn't even get any inflammation to begin with. And this is to emphasize, these animals were 6 weeks when we started, and then we treated them daily for 4 weeks, and then evaluated, and they revert. They look like wild types, versus these ones with a vehicle have severe skin inflammation here. We also see that there is also caspase activation in this case, quantified here, and also RIP3 phosphorylation. So we see a model that at the same time, in the same tissue, in the same animal, we see apoptosis and necroptosis induction in the same time. And that's what we see often, that these cell death pathways are not completely exclusive, but often you see both of them being induced. These sharpie mice, actually the Cpdm mice, are interesting because they develop other diseases, such as eosinophilic esophagitis. So that's the accumulation of eosinophils in esophagus, thickening, and with time gets worse and worse. So in the same mice that we observe skin inflammation and blockade, here we see very little effect with compound. So now RIP inhibitor has some effect, but not statistically significant. So in addition to esophagus, we also look at the liver. Here we see 50% reduction, okay, in the same model. So you see the same animal in one genetic model, in different tissues affected to totally different levels. In skin, complete protection; in liver, partial protection; in esophagus, very little, if any. And when you look at the liver further, you see there is some caspase activation in the liver, but there is no in esophagus. So that tells us if there is a cell death activation, that RIP1 plays a role, and RIP inhibition will be beneficial. If it's just proliferation, then you will not have this benefit, okay? It's very clear here. The other model I want to switch now is in the gut. Here in the NEMO model, if you eliminate NEMO, then you get blockade of this NF-κB-MEP kinase activation, okay? And you activate RIP1. And so now, if you have NEMO, see in a colon, complete messed up colon, and in ileum less so, but still you lose a lot of Paneth cells. However, if you block RIP1 now with a crossing to RIP1 kinase dead mice, then you see you restore all of that, completely healthy, and colon now completely healthy as nothing happened. Okay, and that's not just reflected on the tissue histology. We see there is a lot of cytokine chemokines. They go up in these red symbols, and when you cross NEMO CKOs to RIP1 kinase dead knock-ins, you restore that black and blue, the same symbols. You completely restore that level. And also you see in NEMO knockouts, you lose RIP1, you get RIP1 phosphorylation. When you cross to kinase dead, of course, you lose that. And we have seen that with the compound as well. So now another model that we can see completely replication of the data with a genetic or chemical tool. So now again, you see colitis, ileitis, and if you have compound, you block that very efficiently, and you have a RIP1 kinase inactivation in blue, the same level inhibition, okay? And again, we see activation of apoptosis here with caspase-3 cleavage that is very nicely blocked both genetically or chemically. So we see now that in this case, again, all these pathways combine, and whenever there is cell death and tissue damage, then RIP1 inhibition, either genetic or chemical, can be beneficial. And it's not one of those all or nothing thing, because we used RIP1 heterozygous, and we see partial response. And also we see here that cytokines again get reduced when we block genetically or chemically RIP1, and of course phosphorylation also gets reduced. And as I said, in dose-dependent fashion, so we can dose down from 50 to 15 to 5 to 1, and we see how with less RIP1 inhibition, more damage in colon and ileum. You know, so it really tells you that at least in this model, where NEMO is deleted in gut epithelial cells, we see a very profound effect of RIP1. And so what I'm getting at this is that NEMO is mutated in patients that suffer from IBD and some other skin diseases, But the one that's really important here is the ATG16L1. ATG16L1 is very frequently mutated in patients that have Crohn's disease. And when you have ATG16L1 mutated to this 300A, then it affects PANET cells. PANET cells are very important in gut, especially in ileum, right? Without PANET cells, you don't have proper protection from you don't make alarmist defenses, and you get massive infection and so forth. So we see here now, again, if you delete ATG16L1 just from the gut of these animals, you rapidly succumb to TNF. You know, like within 4 hours, they all die. However, if you introduce RIP1 inhibitor, great protection here. You see from open symbols to closed symbols, great protection. Similarly, you have A20, also mutated in patients with IBD. This is a study from China, a young girl with IBD and has A20 mutation in this zinc finger linker. And again, with no A20, just deleted in the gut epithelial cells, greatly sensitizes to TNF in RIP-independent fashion, as you see from open to closed symbols. And so I believe that one can really identify that a lot of patients suffering from these inflammatory diseases, and now with the sequencing, we can find these patients and really in selective and targeted fashion treat them with RIP1 inhibitors and help them. And so I believe that there is a lot of cases that Gauss inflammation activates RIP1, and then the RIP3 and caspase-8 combined fashion causes inflammatory cell death and tissue damage, as we see in Crohn's disease, ulcerative colitis, and RIP1 inhibitors would help that. And similarly, in skin inflammation, wherever we see tissue damage, but not in psoriasis, which is really proliferative disease, but in lichen planus, Stevens-Johnson syndrome, or toxic epidermal necrolysis, those are diseases where there is a lot of cell death and tissue damage, and this is where RIP1 inhibition could be helpful. And so now, just to illustrate how when TNF goes on the cells, and in combination with IP antagonist, we have now RIP1 will be ubiquitinated. However, if IP antagonist, I showed you earlier, degrades CIP1, RIP1 is no longer ubiquitinated, now moves to the cytoplasm, and now joins caspase-8 and fat. And if caspase-8 is inhibited or not properly activated, RIP1 will activate with phosphorylate, and then you have RIP3 phosphorylation, MLKL, and you have necroptotic cell death. However, if you block that with the RIP1 inhibitor, you block this and you block all the subsequent events, and you eliminate this necroptotic cell death, and that's how we're trying to help patients with this inflammatory diseases. So at the end, I want to acknowledge many colleagues that help here. Eugene work on IIPs, and Tanya, Matthias on the RIP1, and many other colleagues participated here. Vishwan Kim for all these genetic models, Joshua pathology, Snahel chemistry, Manolis for the Nemo mice, and Wihai and Avik for collaboration on BCL2 antagonist. So I'll stop here and take any additional questions.
Thank you, Domagoj.
Super.
I can just jump here for the first question.
Mm-hmm.
So for majority of these inflammatory pathologies that you mentioned here in the second part of your talk, inflammasomes and pyroptosis seem also to have an important role there.
Yeah.
So 2 questions here. First one is, do you think that these pathways could act in a redundant manner? And because of that, in some of them, the inhibition of RIP1 does not work. And the second question is if you are up to date for the clinical trials targeting Gazermin 1 for those pathologies or in some of these models?
So I'm not aware of clinical trials with Gazdermin. I don't think Gazdermin is in clinical trials. But what is in clinical trials or verified is you have IL-1.
Yes.
Yeah, you have IL-1 beta blocking antibody, and you have NLRP3 inhibitor.
P3, yes.
Yeah, and so I think those, because they're all in the same pathway. First of all, it is different to a certain degree. It's a different set of diseases, okay? For example, when we talk about coronavirus, COVID, and actually Sanofi tried RIP1 inhibitors, I don't think they observed anything there. I think actually someone tried NLRP3 inhibitor, didn't see much, but I think this is very good example where gasdermin D would play a role, and this pyroptotic pathway is clearly activated in these kind of things. Second, the other diseases, they have very strong infection-associated pathway would rely on pyroptosis much more. And I think there is definitely a role for pyroptosis NLRP3 inhibitor in myocardial infarction, okay? There is also in IBD and other inflammatory diseases. And it's still open question. I think they're lagging behind, slightly behind where RIP1 is, but not much. And there are a lot of efforts to explore that. And I can envision that one day, I can envision this whole scenario that in the NLRP3 and gasdermin pathway is more important in some aspects, not in the others, right? And so what is lacking there is good way to detect activation of NLRP3 or gasdermin D in human patient tissues. You know, we have this phosphorypin antibody. That's really the most direct evidence there is RIP1 activation. When we use that, when we get that and use that for gasdermin D or NLRP3, then we'll be much better equipped to say in which disease, to what extent, and so forth, this pathway activate. But it's possible that there is certain degree that RIP1 inhibition will capture and some not, and that way maybe combined with NLRP3 inhibitor or gasdermin D inhibitor if it's developed would be beneficial. Absolutely.
That's my point. That's my point.
Absolutely.
Yes. Thank you, Domagoj.
So I think there was somebody that was raised a hand, Abolaji. Can you turn your microphone on?
Good afternoon. Can you hear me?
Yes, I can hear you now.
Okay, so thank you for this lecture. It's also enlightening. And since day one of this course, I've been learning more and more and going back and forth on this issue of necroptosis and tumor and also. But I had this question earlier, and just for clarification sake, I would like to ask again based on what you've also presented. Between RIP-K1 and RIP-K3, which do you think is the major regulatory molecule in necroptosis?
Well, they are both critical components. RIP1 is upstream of RIP3. And the difference is that RIP1 participates in many more pathways and many more processes, where RIP3 is really more focused on necroptosis, okay? RIP3, what it does in apoptosis or inflammation, it really just goes for RIP1. Without RIP1, it doesn't have ability to communicate with other proteins, right? So if you talk strictly about necroptosis, yes, RIP3 is critical, absolutely critical. And you can activate actually necroptosis without RIP1. If you go to LPS and toll-like receptor 4 and TRIF, you can go to RIP3 without RIP1, right? So for that matter, RIP3 could be more critical, but overall both participate. I don't know if I answer your question or.
Okay, all right. Then I have another question.
Go ahead.
You said that while doing the clinical trials that you observed that the RIP1 or RIP3 does not give a positive result in some tissues, while it does in some. So what do you think happened? What could affect this activity or this result?
So, so these were preclinical studies, what I showed. You know, there was no clinical trials that I showed. So just to clarify that, okay? Second, I think the key is if you have inflammatory cell death, okay? when you have tissue damage associated with inflammation and you see evidence of cell death, that's where RIP1 and RIP3 matter. But if you have diseases that are more proliferative, such as psoriasis, in psoriasis, it's really driven by IL-17 signaling, okay? And antibodies from, I think, Eli Lilly and Novartis against IL-17 are 80 to 90% curative, you know, so it just tells you it's a very different setup. And people go with RIP1 inhibitor. GSK went to psoriasis trial with their RIP1 inhibitors. They didn't observe anything. And I'm not surprised. We looked at that. We would never go there because you have to understand biology of disease and you have to understand biology of your target, of your protein, where it plays a role. Just because it can be target doesn't mean it's going to be important everywhere. So I think if it's cell death mediated disease, it has a chance. If it's not, I think very little chance. That's how we see it, at least.
Okay, yeah, thank you. So
Sure.
lastly, I think the last slide you showed, you said you are working on an inhibitor to inhibit RIP1, thereby stopping the development of or the progression of necroptosis. So then I was now reading in one recent paper that discussed necroptosis and also in pathogenesis of disease. Now, specifically to cancer. So there they discussed that if necroptosis can be stimulated, causing this form of cell death in cancer, and then the cancer cells can bring about the release of DAMs, then these DAMs can stimulate the immune system. So now these two idea is kind of conflicting. I don't know if you can shed light to that.
I think that inhibiting RIP1 does not make much sense in cancer. On the other hand, if you stimulate necroptosis, I can envision that you could promote cell death of cancer cells, or maybe even create a more inflammatory milieu for the surrounding tissues. However, I think this cannot be done in a safe fashion because then you would, promote inflammation of other organs, and for that reason, I think from safety perspective, I would not touch cancer with this kind of necroptotic approach. That's my perspective from what I see.
Okay. Yeah, thank you. I'm trying to ask just to get a more understanding because my research focused on the use of this Ubiquinonation also.
Okay.
That's right. Thank you.
Sure.
No, it's important, Domagoj, because we've been bombarded with a lot of information on immunogenic versus non-immunogenic modes of cell death and cancer, how to make the cancer more visible to the immune system, and then necroptosis and apoptosis would be more, interesting compared to the apoptosis. By the way, hello, Doug. Welcome to the room. So it's a good point to touch these questions. It's very important.
Yeah, I think that, yes, if you stimulate, some level of necroptosis, paroptosis, and you have this inflammatory cell death, you could maybe help with fight cancer that way. But, how to do that in a localized fashion, how to do that in a safe manner, I don't think that has been figured out yet. But on the face value, say, yeah, I don't care how I kill cancer cells, apoptosis, necroptosis, as long as you kill them. Well, it doesn't matter because, are you going to cause more damage to the tissues, to the organism, than you're going to save, so it does.
Well, Domagoj, there's some really nice evidence from Peter Vandenbele and from Andrew Oberst, especially in his Science Immunology paper, where if he produces necroptosis in the local tumor environment by transduction using a virus carrying a constitutively active RIPK3, he got dramatic tumor clearance that was dependent on engagement of the immune response. So in principle, of course, this hasn't been done clinically, but in principle, maybe there's some legs there.
Yeah. So, right. So inhibition of RIP1 does not make sense there, but activation of, controlled, localized, in time and space fashion activation could be beneficial. Absolutely. Yeah.
Okay.
Good to see you, Domagoj.
Good to see you. Hey, are you going to Switzerland or?
No, this is the first Cell Death Gordon conference I'm ever going to miss.
Man.
They changed the date. Remember, for so long they were saying the date in May, and then it changed by a few days, and I had a total conflict. So
Oh, I see. That's a bummer. Okay, that's too bad.
I know, I'm bummed out too.
Yeah, that's too bad. Oh, well, I hope to see you at some other meeting then.
Oh yeah, well, I'm going to two cell death meetings right around it. There's one in Crete and one in.
Oh, okay. I'll see you in Crete then. I'm going to Crete, so I'll see you
Fantastic. We'll see each other in Crete.
there. There you go. There you go.
All right, we'll drink Ouzo.
Small world.
So we are jealous of
Yeah.
this.
We are very jealous.
Hello,
Yeah.
Yes,
Karina.
I'd like to go to Crete.
It's very nice in May. Not too hot, just right.
That's right.
I just like to see you and get some beer with you. Doesn't matter where.
Well,
You know.
you
Damn.
just have to move your butt and we'll make it happen.
It's true.
Let's see.
Absolutely.
So anybody has more questions to
To
Domagoj?
Domagoj.
Come on, guys.
Almost my time. Yeah.
We go over.
Yes.
What? Sorry, Lucas.
I maybe have one question, but I don't know if it's like a really dumb question, because with all this talk, I kept thinking that before these classes and with now all this knowledge, messing with apoptosis, as we talked about with blocking BCL2, has consequences for all the system as thrombopenia. And I am, and I was always scared to deal with necroptosis, because I always think it was worse to mess with all these pathways, because the consequence of necroptosis seems to be very better than apoptosis. So do you have some expectations of what we can see if we start to do trials, clinical trials with drugs that influence on the necroptosis pathways, secondary effects?
So the trials that I'm interested is in inhibiting necroptosis, not promoting necroptosis. Okay. So just to clarify that, if you want to talk about activating necroptosis, then you can ask Doug about that. So, but regarding inhibiting necroptosis, I don't think that we expect to see downsides. I think we need to find the right disease and right situation to help patients. I think it should be safe. We know that, from all the preclinical studies with RIP1 kinase, that mice and rats with RIP3 knockouts and so forth, there is a strong a body of evidence that it could be done in a safe fashion, right? So maybe, yeah, I don't know what could be anticipated. Maybe you can affect some kind of viral infection or bacterial infection. That's one possibility. There are studies around that. We have seen in some cases matters, some cases doesn't matter. Most of the trials usually try to exclude infected patients. So we'll see. That's one aspect maybe that could be affected and that we'll have to deal with later.
Domagoj, I just want to go back a little, just briefly, with Karina's questions, because it seems to me that, every time that there is necroptosis, well, first of all, we know that there are these crosstalks between all these regulated forms of cell death, but particularly necroptosis with release of DAMPs should, right away engage the paroptosis program in the neighborhood neighboring cells or something like that. And that's why I want you to come back and comment on, if some of these inhibitors do not work, do you believe that if you add a combination of inhibitors of, pyroptosis or messing, then because it could be like waves, you prevent some necroptosis, but now you don't see much effect because the pyroptosis was engaged. Then you say, oh, okay, I don't have, but it's like a transient things. So could it be done in
He
waves,
answered.
something like that?
Well, so let me
Yes.
give you
Yes.
a quick answer from the theoretical perspective. Yes, theoretically, and in the mouse studies, yes, you would like to block as many pathways because that way you ensure that you're going to block cell death and reduce tissue damage. That's from theoretical. Practically, you will never be able to do a clinical trial with combination before you have anything shown for the single agent activity. You will have to go with the agents and show that are safe and effective as a single agent in whatever disease it is, and then you can envision maybe doing combination trials down the line. So this is theoretically very interesting, practically very far reach.
Yeah, that's a bit of a bummer because, I mean, if you prove that your drug is safe, even if it's doesn't work for every single case of that particular disease, you should be able to try to use it combined.
Oh, absolutely.
Anyway,
absolutely. Yeah.
but it's good to know from the one that, for me, that I'm in the front line of this research.
Yeah.
But the point is that he told us that maybe there is no clinical trial by targeting Gazermin D at the moment, until the moment.
Well,
we can target NLRP3 and we
NLRP3,
target IL-1 beta, but not Gazermin D.
Yeah,
that's fine. I think it's upstream. The most upstream, the better.
Sure. I mean, you can envision using something like a combination RIPA inhibitor with IL-1/6 blocking antibodies or IL-1 beta blocking antibodies to capture some of these combination effects at the moment, and then later maybe with NLRP3.
Yeah. All right, Domagoj, thank you so much.
Thank you again.
No problem. It was a pleasure. Now you're up for a real treat.
Yes.
Future of the world and everything else.
Yes.
Now it's the Star Trek lecture.
Yeah, Star Trek lecture. Yes. Everyone, put your seatbelt on and get comfortable.
Yeah.
Oh, I have to show you the cover. Oh, I can't share my screen yet.
Oh,
okay.
Marcio, can you stop broadcasting and then allow Doug to share the screen?